So, good morning, everyone. My name is Suprita Verma, and today I will be presenting my uh, work on amyotrophic lateral sclerosis, a molecular docking pathogenesis, role of TDP-43, therapeutic relevance of vithapirin A, and molecular docking. So, ALS is a fatal neurodegenerative disorder that specifically affects motor neurons, which are responsible for voluntary muscle movement. In ALS, there is a progressive degeneration of motor neurons located in the motor cortex, brainstem, and the spinal cord. As these neurons degenerate, the communication between the brain and muscle is lost. As a result, the patient experiences muscle weakness, loss of coordination, and eventually complete paralysis. Importantly, sensory functions usually remain intact, meaning patients are often fully aware of their condition. This disease ultimately leads to respiratory failure, which is the most common cause of death in ALS patients. ALS is characterized by gradual and progressive worsening of the motor symptoms. Initially, patient may experience mild muscle weakness or twitching, but over time, this progression to severe muscle wasting and paralysis. The progression rate varies significantly between individuals. On average, the survival period is about 3 to 5 years after diagnosis, although some patients may live longer with supportive care. This variability in disease progression highlights the complexity of ALS and suggests the involvement of multiple and molecular mechanism, making it a challenging disease to treat. Now, ALS is broadly classified into two main types, sporadic ALS and familial ALS. In sporadic ALS, which accounts for approximately 90 to 95% of the cases, occur without a clear family history and has no obvious genetic inheritance pattern. However, in familial ALS, accounting for 5 to 10% of cases and is associated with specific gene mutations such as mutations in SOD1, TARDP, and FUS, and others. Despite differences in genetic background, both forms of ALS share common pathological features, particularly TDP-43 protein aggregation, which is seen in majority of cases. At the cellular level, ALS is marked by severe interconnected pathological processes. One of the key features is protein misfolding and aggregation, where abnormal proteins accumulate inside neurons. And there is also dysregulated RNA metabolism, which affects gene expression and neuronal functions. Additionally, defective axonal transport disturbs the movement of essential molecules along nerve fiber, leading to neuronal stress. Finally, persistent neuroinflammation further exacerbates neuronal damage. These combined mechanisms ultimately results in the motor neuron degeneration and cell death. One of the most important molecular hallmark of ALS is TDP-43 or DNA binding protein of 43 kilodalton. Under normal condition, TDP-43 is now TDP-43 is primarily a nuclear protein involved in RNA splicing, RNA transport, and gene regulation, all of which is essential for neuronal damage. However, in ALS, TDP-43 undergoes abnormal modifications such as phosphorylation and ubiquitination. It becomes mislocalized from the nucleus to the cytoplasm, where it forms toxic protein aggregates. These aggregates are found in approximately 95% of ALS cases, making TDP-43 a central driver of disease pathology and a highly attractive therapeutic target. Now, molecular docking is a computational technique which is used to study how a small molecule interact with our target proteins. It helps to predict the binding affinity, binding orientation, and also the stability of the protein and ligand complexes. Docking is widely used in early stage drug discovery because it significantly reduces both time and experimental cost. In ALS research, molecular docking allows us to screen potential inhibitors against targets like TDP-43 and identify compounds that may stabilize protein structure or prevent aggregation. Thus, docking plays a very crucial role in rational therapeutic designs. Vithaferin A is a natural bioactive compound derived from the Withania somnifera, commonly known as ashwagandha. It belongs to the withanolide class of steroidal lactones and has been used in Ayurvedic medicines too. Biologically, vithaferin A exhibit multiple beneficial properties, including anti-inflammatory, antioxidants, immunomodulatory, and neuroprotective effects. In neurodegenerative disorders, vithaferin A has been shown to suppress a necrosis factor kappa B mediated inflammation, enhance autophagy, and promote clearance of misfold proteins, making it a promising candidate for ALS research. Now, for docking studies, TDP-43 protein structure with PDB ID 5W52 was selected due to its high resolution of 1.4 angstrom, ensuring structural accuracy. Then the protein was retrieved from the RCSB protein data bank, while vithaferin A was obtained from the PubChem database in 3D SDF format. Then protein preparation involved removing heteroatom, adding polar hydrogen, and saving the clean structure. Ligand protein preparation included energy minimization and conversion into AutoDock format using PyRx and OpenBabel. The docking was performed using AutoDock Vina, where the grid box was carefully placed to cover the entire protein. Multiple binding poses were generated, and the pose with the lowest binding energy was selected for analysis. Docking analysis revealed a binding energy of minus 4.9 kilojoule per mole for the interaction between vithaferin A and the protein 5W52. This value indicates a stable but moderate interaction. In docking studies, more negative, the binding energy corresponds to stronger interaction compared to high affinity inhibitors, and this result suggests that a preliminary or exploratory binding events. However, it is important to note that the natural compounds often exhibit moderate binding affinities, yet exert significant biological effect through multi-target and synergistic mechanism. Therefore, vithaferin A may act as the potential, but it holds promise as a lead compound that can be further optimized through chemical form modifications. This is how I have done the analysis of my work. And to conclude, ALS is a complex neurodegenerative disease with limited therapeutic options. Then TDP-43 plays a central role in ALS pathology, making it an important drug target. Though molecular docking, vithaferin A demonstrated a moderate but stable interaction with TDP-43 protein structure 5W52, supporting its potential as late compound. Further studies, including molecular dynamic simulations and experimental validation, are required to confirm its therapeutic relevance. Thank you so much.